Tuberculosis vaccines, from traditional to modern. Traditional vaccine, BCG, Bacillus comment, Garin. Type/technology. Live attenuated bacterial vaccine. Derived from Mycobacterium bovis, closely related to M. tuberculosis. Developed by Albert Comet and Camille Garin in 1921. It is the only licensed TB vaccine currently in use worldwide. Mimics infection, activates macrophages and induces cell-mediated immunity, Th1 response. Stimulates IF and gamma, producing T cells. Prevents severe TB in children, miliary TB and TB meningitis. Inexpensive and safe for newborns. Limitations. Poor protection in adults against pulmonary TB. Variable efficacy, 0 to 80%, in different populations. Can cause infection in immunocompromised individuals, example HIV+. Recombinant BCG vaccines, genetically modified. Modern biotechnology has improved the classical BCG strain to enhance its immunogenicity and antigen expression. Vaccine. Technology. Feature/target. rBCG 30. Recombinant BCG overexpressing AG85B. Enhances Th1 response. VPM 1002. BCG modified to express Listeria lysine O, LLO, from Listeria monocytogenes and URC deletion. Improves antigen presentation via MHCI rightward arrow stronger CD8 T cell response. Errors 422, HLY. Similar to VPM 1002. Better cross-presentation of TB antigens. To make BCG more effective for adults and safer for immunocompromised populations. VPM 1002 is in phase 3 trials, most advanced recombinant BCG. Subunit vaccines. These vaccines use purified antigens or fusion proteins from M. tuberculosis combined with a strong adjuvant. They are safe, defined, and stable, but less immunogenic without adjuvants. Vaccine. Antigens used. Adjuvant/platform. Status/highlights. M72/S01A. MTB32A plus MTB39A fusion protein. S01E, liposome plus MPL plus QS21. Phase IIB success, 50% protection in adults with latent TB. H56IC31. AG85B plus ESAT6 plus RB2660C. IC31, TLR9 agonist plus polycationic peptide. Phase 2 trials. H4IC31. AG85B plus TB10.4. IC31. Phase 2 trials. ID93 plus GLASE. Fusion of 4 antigens, RB2608, RB3619, RB3620, RB1813. GLASE, synthetic TLR4 agonist in oil emulsion. Phase 2 trials. Hybrid I/hybrid 2. Multi-antigenic fusion proteins. CAF01 adjuvant. Early phase. Recombinant protein expression, usually in E. coli, purified, and formulated with synthetic adjuvants to elicit Th1 response. Viral vector-based TB vaccines. These use genetically engineered viruses, harmless, to deliver TB antigens into host cells inducing strong T cell immunity. Vaccine. Vector used. Antigen, S. Purpose/stage. MBA85A. Modified Vaccinia ankara, attenuated Poxvirus. AG85A. BCG booster, tested in phase IIB, safe but limited efficacy. Ad5 AG85A. Adenovirus type 5. AG85A. Strong CD8 T cell response, intranasal version under study. CH ADOX 1.85A. Simian adenovirus, CH ADOX 1. AG85A. Oxford developed, heterologous prime, boost. ADHU 5 AG85A. Human adenovirus 5. AG85A. Induces mucosal immunity. Molecular cloning of antigen genes into viral vectors, production in mammalian cells, purified as vaccine. Live attenuated M. tuberculosis vaccines, next gen. Instead of M. bovis, BCG, these use genetically weakened M. tuberculosis itself, keeping antigen similarity higher. Vaccine. Modification. Description/stage. Vac. Deletion of FOP and FAD-D26 genes. Derived from M. tuberculosis H37RB, retains broad antigen repertoire, safe in animals and humans, phase 3 underway. HLY. BCG expressing Listeria lysine. Improved antigen presentation. Achieve better immune mimicry of real infection but with safety ensured by gene deletions. Genetic engineering and molecular attenuation of virulent M. tuberculosis. DNA vaccines. Contain plasmid DNA encoding TB antigens. After injection, host cells express the antigens, T cell activation. Vaccine. Antigen, S. Description/stage. DNA AG85A. AG85A. Induces both Th1 and CD8 T cell response. DNA ESAT6/CFP10. ESAT6, CFP10. Targets early secreted proteins. DNA HSP65. Heat shock protein 65. Shown protection in mice. DNA AG85B ESAT6. Fusion construct. Enhanced cellular immunity. Recombinant plasmid DNA with eukaryotic promoter rightward arrow delivered via needle or electroporation rightward arrow antigen expression by host cells. mRNA-based TB vaccines, cutting-edge technology. Inspired by the success of COVID-19 mRNA vaccines. Vaccine. Antigen, S. Platform. Status. mRNA 1647 TB, Moderna. Encodes MTB antigens, example AG85B, ESAT6, CFP10, PPE family. Lipid nanoparticle, encapsulated mRNA. Preclinical/early phase. CureVac TB program. Multi-antigen mRNA vaccine. LNP-based mRNA. Early development. Synthetic mRNA encoding TB proteins packaged in lipid nanoparticles, LNPs, rightward arrow translated in host cells rightward arrow T cell activation, both MHC-I and II. Fast to design and produce. Non-infectious and scalable. Strong Th1 type and cytotoxic T cell immunity. Peptide and epitope-based vaccines. Contain synthetic short peptides, epitopes, that mimic T cell targets. Identified via immunoinformatics/reverse vaccinology. Vaccine. Type. Description. T-epitope mix. Peptide cocktail. Computationally predicted epitopes. Multi-epitope fusion peptide. Recombinant polyepitope protein. Induces IF and gamma, producing T cells. Synthetic chemistry plus bioinformatics to identify MHC binding epitopes. Combination/heterologous prime, boost strategies. To maximize immune memory and breadth. Prime with BCG or rBCG. Boost with subunit/viral vector/DNA vaccine. Example. Strategy. Benefit. BCG prime plus MBA85A boost. Classical plus viral vector. Expands T cell memory. BCG prime plus H56IC31 boost. Live plus subunit. Enhanced Th1 response. VAC prime plus M72 boost. Next gen combination. Broader protection spectrum. Era. Vaccine. Platform/technology. Status/comments. Classical. BCG. Live attenuated M. bovis. Licensed since 1921. Recombinant BCG. VPM 1002, rBCG 30. Genetically engineered BCG. Phase 3. Live attenuated MTB. VAC. Attenuated M. Tuberculosis. Phase 3. Subunit. M72/S01E, H56IC31. Recombinant protein plus adjuvant. Phase IIB, 3. Viral vector. MBA85A, Ad5AG85A. Modified vaccinia/adenovirus. Phase 2. DNA vaccine. DNA AG85A, DNA HSP65. Plasmid DNA encoding antigen. Preclinical. mRNA vaccine. mRNA 1647 TB. LNP encapsulated mRNA. Preclinical. Peptide vaccine. Epitope-based constructs. Synthetic peptides. Preclinical. Prime, boost. BCG plus MBA85A, H56. Combination approach. Ongoing trials. Key technological evolution. Generation. Core technology. Immunity targeted. First gen, classical. Live attenuated, M. bovis. Broad innate plus CMI. Second gen. Recombinant BCG/subunit. Defined antigens, safety. Third gen. Viral vector/DNA/RNA. Cellular immunity, memory T cells. Fourth gen. Epitope/multi-antigen. Precision immunity and broad coverage. Tuberculosis vaccine development has evolved from the simple live attenuated BCG to advanced genetically engineered, subunit, DNA, viral vector, and mRNA platforms, aiming to induce potent, durable, and adult protective cell-mediated immunity.